sustainable drug manufacturing. My name is Diana Castro and I am Senior Content Producer at Informal Markets, and I'll be moderating this session today. Just a little housekeeping before we get started. Remind you that this is a live session, so if you have any questions in the presentation, please type them into the question box in your control panel. Please feel free to submit your questions throughout the session, and I'll put as many questions as possible to our speakers at the end. If you have any question that you would like to address or direct to a specific presenter, please put the name of the presenter at the beginning so I can directly address that question to the person. So here is the agenda and our speakers for today's session. This webinar will provide an overview of the challenges development, as well as some of the key trends and examples on initiatives driving green chemistry and sustainable drug manufacturing. Before we get started, Informa is trying to understand how sustainability is evolving within the pharmaceutical landscape. And we will have 3 short poll questions along the session. One now, another after our first presenter concludes, and the last one before the Q&A. So therefore, I'd like to kick off this session with our first poll question for all of you. I'm going to read the question out loud. So do you believe the pharma industry is undergoing a shift towards a more sustainable practices? And the answers can be either yes, no, or somewhat. I'll leave you a few seconds for you to reply to this question, and you should be seeing a popping up within your screen, so you can choose one of the answers, and so that it will help us understand how sustainability is evolving within pharma. I'm gonna check the answers. You can see in the screen. Thank you for all of those replying to this. So without further ado, I'd now like to introduce our first presenter, Julien Boutet. He is the Innovation Manager at Seqens, where he is responsible for developing the selection methodology of future proprietary APIs, as well as responsible for enhancing the value of Seqens key technologies such as biocatalysis and flow chemistry. Now I'll hand the session over to you, Julien.
Yes, hello. Normally you can all see me and hear me. So thank you to the organizing committee to invite me to present this session today. Thank you all for joining this conference. So I would like to take the opportunity today to share with you some advantages of biocatalysis in API manufacturing drugs, especially that this technology really clearly met the many criteria of green chemistry. First, I would like just to introduce shortly Seqens. Seqens is a global leader in pharmaceutical industry and specialty ingredients, roughly with a ratio 7:3 in terms of EBITDA. We are present all over the world with more than 3,000 employees. We have 24 manufacturing sites all over the world and more than 1,000 customers. Within Seqens, there is 3 R&D centers, one based in Canada and two based in France. One of them in France is Protéus by Seqens. This R&D has been created in 1998 and acquired by Seqens in 2017. It's based in Nîmes, south of France, with 25 employees. The expertise of this R&D group is really the enzyme. It's based on 3 pillars. The first one, as you can see on the left of the slide, is identify new native enzymes. So this is based on the collection of strains, and more than 1,000 has been sequenced. That means that we know which activities there is inside based on the enzyme. Second pillar is having the expertise to improve an enzyme with some technology that we call direct evolution. So you start from a native enzyme with specific activity and you will increase the performance. The third pillar is having the capacity to scale up, to produce the enzyme, to scale it up from gram scale to ton scale, which match perfectly with Seqens, because then we can do chemical reaction using biocatalysis within Seqens. So today that makes Proteus the only European-based company which offer both direct evolution of enzyme services and large-scale reaction capabilities. Now, let's go inside the topic. So first, I would like to maybe remind everybody, what is an enzyme? An enzyme is a protein. It's a protein which is produced by a microorganism. This is a biological catalyst, which means that in nature, enzyme exists as a catalyst to speed up some biochemical reaction. When I say speed up, it can be up to 1 million times ratio. So this is really specific. The scheme you see on the top right is that you will see that enzymes are really specific for one task based on one molecules. And last important information is enzymes are so specific that if you change one amino acid of the structure, you might change completely the conformation of the protein in terms of 3D conformation, and then you can change completely the activity or the specificity of the enzyme. How does it work? Usually it's in 4 steps. The first step is the substrate will arrive close to the enzyme. Why? That's why the 3D conversion is important. Then the 2 substrates will be grabbed by within what we call the active site, and then the third step is this chemical reaction will happen. And then you have enzyme product complex. The last step is the enzyme which releases the product. So if you are wondering, are the enzymes present in the market, the answer is clearly yes. There is many enzymes which have been developed and which are commercialized in lots of areas. If I take 3 key examples, in cheese production, in bread production, in beer production, all the enzymes are used at industrial scale. Another example is what I want to mention, because I've been working on this area, is animal feed area, where enzymes are used to help, for example, poultry to digest better what they're eating by degrading some carbohydrate, for example. Now, the question is, can we use enzyme for chemistry? And the answer also is yes. We can use enzyme as that what we call biocatalysis transformation. When you take what I call here product A, and you want to do hydrogenation, usually in chemistry, you will think, okay, I will use a palladium catalyst. I will add hydrogen, put under pressure, and I will do the product, which will be an alkene, which is hydrogenated. But in fact, now with many enzymes which have been developed, it exists sometimes we can do this transformation. And if you look at the top right, this example is a well-known example because it's a key intermediate for levodopa, which is an API. Nowels has developed what we call a catalyst based on rhodium with chiral ligand, and which are able to do this hydrogenation with enhanced selectivity. It means that you will create a chiral carbon close to the amino acid. But this typically now enzymes are known to do the same kind of transformation. And what we did in Proteus, we developed and we commercialized what we call sequencing kit. What they are, that's 7 families of enzymes, which are specific for 7 kind of chemical reaction, and they are on the market in order for any chemist who is working on development, who is working in finding new routes of synthesis, can test directly in his lab this enzyme for specific reaction. So when you think about some rhodium catalyst and you have in your lab several catalysts at your disposal, you can do the same with enzyme. So this is the purpose of having this kit available for any chemist. Now, the question is enzyme or biocatalysis respect the green chemistry principle? And here I just mentioned on this slide what are the 12 green chemistry principles which have been introduced in 1991. And if you look at just roughly, I didn't go too much into details, but if you look at what enzyme can do, when you look at designing safer chemicals, clearly biocatalysis will help on this, because most of the time the reaction will be done in water and not in organic solvent. Can we do more efficiency in terms of energy? Yes, also. I mentioned the hydrogenation. You will need in typical chemistry to do under pressure. Enzyme will work at room temperature, at pH 7, and atmospheric pressure. Can you save some carbon? Yes, also, because biocatalysis will not need to do what we do in chemistry. I protect a group, I react on another one, and I deprotect the first one. Usually, as they are so specific, enzyme can do directly without protection. So for example, if I take another example, will I save some other chemicals? Yes, clearly, because this is proteins, this is natural, it's only amino acid. Also, how do you produce enzyme? You produce enzyme just by using oxygen and glucose. So clearly, really easy raw materials. So and so on and so on. You can see that most of this principle matches biocatalysis. Now, I would like to share with you some examples that we have developed within Seqens. The first example is deserine. Deserine is a non-natural version of the amino acid serine. It is used, and I mentioned here 3 API, which are already commercialized in the market, and that you need this specific intermediate. We developed a chemical process using Seqenzyme AL, which is an enzyme, and able to add the formaldehyde on the glycine and obtain only one form, the deserine. So this process now is running every month within Coutern site of Seqens. And some key data, we produce more than 10 tons per year of these intermediates. We are using for the enzyme production 15 cubic scale fermenter. So really, this is really high scale. And in terms of chemistry, is standard chemical reactors. We use 5% of enzyme. We have really high selectivity. We do not need any protecting groups. And if you look at the enzyme residue in the final product, this is under detection limit. So it's perfectly matching any requirement that we'll need in pharma. And most important is, okay, is it competitive? But if you look at the cost impact of only the biocatalyst on the production of deserine, we are around below 6%. Second example, I cannot share with you the reaction, but it's also an intermediate which is present in 3 also different API, antivirals with really high market business. It's also done at our site at Coutern, and the difference is not in terms of scale, because now we produce more than 4 tons per year. The difference is that this biocatalyst is produced within Proteus, So, and why? Because we only use this time 400 ppm. So really a little amount of this enzyme. Why? Because it's really specific for the reaction we want to do. So higher activity. And if you look at the final cost impact, again, it's below 8%. So when you look at that, the first takeaway message I would like you to take today is that whenever you use a biocatalyst, enzyme, typically in our case, within Seqens, represent less than 5 to 10% of the overall production of one single step. And this is true in pharma, but it's also true in food and large-scale chemical ingredients that we have done within Seqens. But if it's still too high, you have some options to still do improvement by what I mentioned early, enzyme evolution. We can have again and work to have more efficient enzyme, which will again decrease your cost impact. Some ideas that people have when we talk about enzyme is that enzyme are expensive. This is wrong. This is a wrong idea, because in fact what matters is the quantity and the cost. If you use a little bit of really expensive product, still the cost impact will be still low. Second idea that we have is that we believe that enzyme production, so catalyst production, is not robust and difficult. Today, fermentation processes are now well known. If you have a good chemical engineer, process engineer, you will be able to do the scale up without any issue. The same as we do in chemistry. Third one is, does it take too long? Process development is really long, and sometimes you miss opportunities. That is now not true anymore, because now there is lots of work has been done, and we are able to scale up a process of this kind of biocatalyst within 6 months. And we have done it in parallel to other API projects, and this was not the critical path of the development. So now I would like to go in one specific area that the enzyme, I believe, will be really interesting in the future, is metal replacement. So here is 3 schemes showing the palladium, the rhodium, the ruthenium price over the last 5 years. And if you look at the rhodium price, the increase is pretty impressive. So this is not due to COVID. You have a small decrease in last year, but this is not the reason. The reason is that a growing demand, especially for automobile, which makes that the chemistry is not the main market. And even though we know that in chemistry, we recycle the catalyst over roughly 75 to 80% by some process, still, this cost will be impacting your overall competitivity of your process. So that's why I believe enzyme could be an alternative, because you will see that the price is less volatile than metal. So again, I will go more into details, and I will give an example of an API that we studied within Seqens last year. So this API has been marketed since 2018. It's called Tanoprevir. It was developed for treatment of HCV. But last year it was tested in March, April 2020 on a combination with ritonavir to treat COVID-19. So we just, for the exercise, wanted to know, okay, how we approach this API. Can we use biocatalysis in this API? So if you look at this building block of this API, we have seen that the process today use rhodium catalyst. So, okay, can we do and replace it by an enzyme? So when we go into details, this is a rhodium catalyst. So you can see that you have a chiral ligand, so the catalyst will be expensive, and rhodium with the price, you will see the impact. So again, I only show the price until 2020 to not show, but it has continued to increase the rhodium price. And can I use enzyme? Yes, clearly, because in the literature, there is a family of enzymes that called N-reductases, which are able to do this kind of transformation. So make a hydrogenation, enantioselective, creating the chiral ligand at the right position. And we went to the lab, so we used our E-Red kit. Within 1 day, we developed a quick method to see the conversion, and within another day of work, we could test the 10 enzymes. And here are the results. So one enzyme, E4454, can convert the raw materials into the expected product, with 26% of yield. So this is a proof of concept. After, what you need to do before to go further and to develop the process to make this enzyme is, okay, are we competitive? And this is pretty easy to do. Just need to compare in terms of cost. So here I show you a graphic showing the comparison between rhodium with the price of 2020, rhodium with the price of 2018, the yellow curve, and the enzyme price. So I took some hypotheses, simple hypotheses. So I have the same chemical yield. The use of metal or enzyme is based either on literature for the rhodium, or 3% of enzyme, or 0.5% of enzyme. What I count in the cost, the metal, I only take in consideration the metal cost. I do not consider the ligand. I do not consider how to make it in order to favor this solution. And I compare with biocatalysis, where I check the price of the enzyme and everything which goes to do the conversion. And clearly you can see that, yes, based of 2018 cost of rhodium, the enzyme will be difficult to be competitive. But if you look at the cost of 2020, clearly within a low volume already, you will be able to be competitive. And what is also interesting to show is that it doesn't matter the quantity of enzyme. At the end, the cost will be similar, because you will go in large-scale fermenter, and then at the end, as I mentioned, it's glucose, oxygen, the cost will not be changed. So in this particular case, we can say that based on the price of 2020, biocatalytic process is 4 times cheaper than a rhodium process. So keep in mind that metal catalyst price is volatile price, which is not the case for biocatalyst. Operating conditions are more smooth in the case of biocatalyst, with pH 7, room temperature, atmospheric pressure. And after, in pharma, the key point is to also make sure that you have removed the catalyst at the end. And rhodium can be an issue. There is some toxicity. Protein can be also an issue to remove, but you have less risk of toxicity because that's only amino acid. So that's how we work within Seqens. So first step, we need to find what we call a hit. So this is really important. We need to find that, okay, the conversion we want to do can be done with an enzyme. So we have available some Seqenzyme kits that any chemist now can test directly in his lab if he wants. If he needs some expertise, of course, you can always contact Proteus, and we have now experiences to be able to help you. This is important to have the starting point, and then you can do a cost evaluation. Can enzyme be competitive, as I mentioned you on the case of Danoprevir. After, you need to decide, is it worth it or not to go further? And after, there are different options. So I mentioned here what we call Sequencing Explorer. Let's imagine that you have a hit, but okay, it's not competitive enough on the paper. Then you will need to look at other enzymes. And then we have library with enzymes that we know that will be able to match with your substrate. Another option is that, okay, we have pretty good conversion, but it's still not enough. We need to have, I don't know, an enzyme which is thermostable at higher temperature. We need to have a better conversion. Then what we can do is what we call Seqenzyme Builder, which is a direct evolution, really specific to enzymes. So we can target one amino acid as part of the amino acid, and then this can be done in really high throughput screening to test lots of enzymes in a short time. And always the purpose is to get a better hit and better conversion. These two steps are also really important if you want to create IP. Of course, you want to possibly protect your innovation, and then to make an IP, you will have to have different experiments on different enzymes. The last step, of course, once you know that you can be competitive between a former process or especially a new process, then you can go to the next step, which will be fermentation process, optimize the catalyst, the production of the catalyst, and in the meantime, the chemical process, so the biocatalytic process, where you need also to optimize it. And at the end, you can, at each step, compare the competitiveness of the process and the efficiency in terms of cost. So some key takeaway messages. So today at Sequens, as I mentioned you, the cost of the biocatalyst represent around 5 to 10% of the overall cost. The main uses of biocatalysis are chiral molecules, steroids, as I mentioned you, replacement of metal catalyst due to high increase. And the last point is that in terms of pharma, if you look at pharma application, usually a 300-liter fermenter is enough to have the quantity needed for some APIs. So that means that we can directly do it within Proteus, so that you will shorten your time of development. Yeah. That's what I wanted to plan today. So thank you for your attention.